Okay, this lecture is the first in a series of lecture for the advanced cell biology students, looking at how protein transport occurs within the cell and within different cellular compartments. So after this session you should be able to explain the main mechanisms of protein transport within the cell and be able to distinguish between different cellular compartments within the cell and relate the mechanism of protein transport used to that particular compartment, and also discuss how proteins undergo transmembrane transport. So this is going to be the basics of all of these, and then in subsequent lectures we'll look at each transport mechanism in more detail. So this slide shows all of the different places that proteins can be transported within the cell. And we're starting off with proteins in the cytosol because that is where the ribosomes are, that is where proteins are made. So starting from the cytosol, proteins can get to nucleus, mitochondria, endoplasmic reticulum. Don't really talk about plastids in this module, and also peroxisomes. Don't really talk about those all that much. I'm mostly going to be talking about nucleus, mitochondria and endoplasmic reticulum. So for the first of these is cytosol to nucleus, and this is what we call gated transport. And this is because the nucleus and the cytosol are separated by a double membrane called a nuclear envelope. And for proteins to get from the cytosol to the nucleus and back again, they go through a nuclear pore. Now this is not transmembrane, it is called gated transport, and we'll see why shortly. So to get from the nucleus to the cytoplasm, the proteins don't actually cross a phospholipid bilayer. They go through a gate or a pore within the double membrane, and as we'll see on the diagrams, this is distinctly different to transmembrane transport. So that is cytosol to nuclear, which is gated transport. We've then got transmembrane transport. So this is where a protein is threaded through a pore in a plasma membrane so that the protein directly crosses a phospholipid bilayer. Now to do this, proteins are unfolded and pass through with no secondary or tertiary structure. That's different to gated transport, where the proteins go through as fully folded functional proteins. And for the transmembrane transport, typically we've got proteins going from cytosol to mitochondria, cytosol into the endoplasmic reticulum. They're the two main ones that you will be dealing with. We've also got the vesicular bound system. This is everything shown in green, and this is dealing with proteins. Once they've got into the endoplasmic reticulum, they can be passed on from vesicle to vesicle to any other of the vesicle bound system. Not only that, you've got the opportunity of cells getting to from the exterior into the cell. So this is from proteins taken up from outside the cell into the cell, into an endosome, and then once they're in an endosome they can go to any part of the vesicle bound system. Now what you'll notice is that this is effectively mostly a one-way arrow. There are very few situations where proteins get out of the endoplasmic reticulum. They get in, they tend not to get out of the vesicle bound system, but there are a few examples, and particularly misfolded proteins can be exported out. But I'll talk about those in the proteasome lecture next semester. Okay, so looking at how proteins are made in the cell, mRNA, ribosomes and nuclear proteins have to be transported via the nuclear import and export transport system. So if you think about what it would take to create a new protein, you need transcription factors to get into the nucleus. You need RNA polymerase to be in the nucleus. You need the mRNA to be able to get out of the nucleus. You need ribosomes. Ribosomes are put together in the nucleus and exported as large subunits. So you need protein import into the nucleus. Those proteins link up with ribosomal RNA, and then they're exported as intact ribosomes from the nucleus. So nuclear to cytoplasmic cytosol shuttling is absolutely crucial for all stages of protein synthesis. Once we're at the point of having an mRNA exported from the nucleus to the cytosol, new proteins can then be made in the cytosol for sort of free proteins by the free ribosomes or the endoplasmic reticulum bound ribosomes. So the endoplasmic reticulum bound ribosomes are crucial for secreting proteins directly into the ER, the endoplasmic reticulum. So anything that's due to be secreted from the cell is mostly made by a ribosome attached to the ER, and as protein synthesis occurs, that polypeptide chain is threaded through a translocation channel into the endoplasmic reticulum. So those are your ER-bound ribosomes. And then we've got the free ribosomes, which make proteins that are free, floating around in the cytosol, but also proteins that need to get to the mitochondria, proteins that need to get to the nucleus, and some proteins that need to get into the endoplasmic reticulum. So not all ER-destined proteins are made by the ER-bound ribosomes. Some are made freely in the cytosol by the free ribosomes, and then they make their own way to the endoplasmic reticulum. So once proteins are made, they have to get to their destination. They have to get to the right organelles. And proteins have signal sequences on them that send them to the right place. So proteins that are produced by the free ribosome cycle in the cytosol will have signal sequences. These are short peptide sequences that get them to the right place. So a protein that needs to be in the nucleus will always have lysine, 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 arginine, lysine in an accessible part of the protein, and that signal sequence gets that protein into the nucleus. Equally, if it's a protein that needs to get out of the nucleus, it has a slightly longer, more complex signal sequence. To get into the mitochondria, we have this very long N-terminus sequence here. And to get into the endoplasmic reticulum, we also have another N-terminal sequence as shown here. Now, there are very few of these that you need to remember off by heart, but I would recommend remembering the nuclear import signal sequence and some of the ones that I'm going to talk about in the endoplasmic reticulum lecture. These long ones, you do not need to memorize those signal sequences. Now, one thing to note is if a protein is going to be resident in the cytosol, it will have no signal sequence. It will be produced by the ribosomes in the cytosol and it will stay there. So this slide illustrates just how powerful signal sequences are for protein shuttling, and this is an image of staining of the large T antigen. That's a viral protein, and the large T antigen normally exists within the nucleus of cells. It's produced in the cytosol by the ribosomes, and it has to make its way to the nucleus, and it has a nuclear localization signal: lysine, 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 arginine, lysine. So we can see the protein is in the nucleus. You can't see the cytoplasm because there is undetectable levels in the cytoplasm. Conversely, this experiment has been done where the large T antigen has been mutated, where this threonine here has been introduced where there is previously a lysine. The effect of that is we can now stain for this protein and see the outline of the protein packed in the cytoplasm with effectively no protein in the nucleus. And so this illustrates that by changing one amino acid of one signal sequence, we go from this pattern of it all going into the nucleus to this pattern of the protein being made in the cytosol. This protein has no functional signal sequence, so it stays in the cytosol, and we can see the outline of the nucleus, and inside there is no protein present. So this is purely an illustration of how powerful those signal sequences are, and then we'll cover the nuclear import-export pathway that uses this signal sequence in a subsequent lecture. So I'm now going to cover the basics of transmembrane protein transport system. So in this system, proteins are transported from the cytosol to the mitochondria, endoplasmic reticulum, and in plants, the chloroplasts. So almost all of the proteins present in mitochondria and chloroplasts are made by the free ribosome cycle, and those proteins have to make their way to that destination. So to get there, they have to have a signal sequence on them. These protein signal sequences are recognized by transmembrane proteins on those destinations, so on the endoplasmic reticulum, also on the plastids, on the mitochondria, and they will help those proteins get across that transmembrane, across the plasma membrane, to their new destination. So you should be familiar with this from your CBBM lectures last year, but I'm going to remind you of it anyway. This is the free ribosome cycle. So this is where an mRNA is picked up by a ribosome, and then that ribosome will allow translation of that mRNA, and we get an extending polypeptide chain coming off that ribosome. So we've got the free ribosomes and the endoplasmic reticulum bound ribosomes down here, but they come from a common pool of ribosome subunits which are lurking around in the cytosol. These proteins are produced freely floating in the cytosol, and then they can make their own way by transmembrane transport to the nucleus, by transmembrane transport or other mechanisms, so mitochondria and ER is transmembrane, or they could do gated transport to the nucleus. In contrast, endoplasmic reticulum destined proteins are made by the same ribosomes, but as the mRNA is translated and the peptide sequence starts to emerge from the ribosome, an endoplasmic reticulum signal sequence is the first thing to be sort of produced. That will link up with a signal recognition particle, or SRP, and that SRP will bind to the newly emerging peptide and pause translation. This SRP will then bind into a signal recognition particle receptor here, and that will bind onto the SRP, the signal recognition particle. So we've got a mRNA which has been translated, but it's been paused, bound to the SRP, bound to the SRP receptor on the surface of the endoplasmic reticulum. What then happens is this signal sequence interacts with a translocation channel, which is this Sec61 channel. This arrow should be up here. So that interacts with the Sec61 channel, and then this allows translation to restart, and then the polypeptide chain is threaded through that Sec61 channel. So it requires ongoing translation to occur to have that protein pass straight through that channel into the endoplasmic reticulum. This is just shown in more detail here. Here is the two ribosomal subunits. There's the mRNA. This is the peptide that is being produced. We've got tRNAs coming into the ribosome, each with a amino acid residue on there, and that is resulting in the extending polypeptide chain. As this signal sequence emerges, that binds to the signal recognition particle at point 1. At point 2, the signal recognition particle is bound to the emerging signal peptide, paused translation, and then bound into the signal recognition particle receptor. This allows the release of the SRP, and then this signal sequence here can interact with the Sec61 complex, open that translocation channel, and translation can restart. Then the protein is, the mRNA is translated more, we get an extension of the polypeptide chain, and the peptide is threaded through that Sec61 channel. So that process here works for peptides for the endoplasmic reticulum bound ribosomes. However, as I mentioned, some proteins make their own way to the endoplasmic reticulum and are made by the free ribosomes. So this is what we call a post-translational system. So it doesn't always require ongoing translation. On the previous one, you needed ongoing translation, whereas proteins can get there post-translationally. That is, they are produced in the cytosol, they make their own way there after translation is finished, and then enter the endoplasmic reticulum. So here we've still got Sec61, that complex that we saw previously. The endoplasmic reticulum peptide signal sequence can open that complex, and there's a whole bunch of other proteins linked in with it as well now, so this is a more complicated system. So we've got Sec62, 63, 71, 72 and other proteins around. We've also got these what are called binding proteins. We'll call them chaperones as well. These will help pull through this polypeptide chain through that pore in an energy dependent way. So these are often called binding proteins, also called chaperone proteins, also called heat shock proteins. So this is a whole family of proteins that help transport proteins through these translocation channels, but also to help refold them. And we'll come across heat shock proteins in the proteasome lecture next semester. So at this point we have our protein in the endoplasmic reticulum. Those heat shock proteins will help to refold that protein into its correct configuration, because here it is just, there is no tertiary structure or secondary structure really, it's just a polypeptide chain. So what I'm now going to cover is the outline of the vesicular transport system, as what can happen to that protein once it's in the endoplasmic reticulum, and where can it go. So once in the endoplasmic reticulum, the protein can be passed on to the Golgi. So in the next session or next week we'll cover endoplasmic to Golgi transport, and we'll cover how vesicles form, fuse and are transported from one place to another. Once they're in the Golgi, the proteins can be exported to the cell's exterior in secretory vesicles, or some proteins can be sent off to other structures such as the lysosome for destruction. We'll cover that in some subsequent lectures as well. We'll also cover endocytosis, which is using the vesicle bound system to bring proteins from outside the cell into the cell, and then those proteins can be used intracellularly. So we'll cover that in the coming weeks. So everything to do with the ER and the Golgi transport, lysosomes, functions of lysosomes, how proteins get from A to B in a vesicle, what sort of tags they have, what sort of tags the vesicles have. We will cover that in the next few weeks' lectures on the protein transport series of lectures. And just highlighted on here is the paths for destruction of proteins. So some proteins will go to the lysosome for destruction, particularly sometimes things which are endocytosed, so brought in by endocytosis into an endosome. From there they can end up into a lysosome. And then highlighted in blue is the exocytosis pathway, which we'll cover in the next few lectures, which is proteins made by ribosomes, passed on to the endoplasmic reticulum somehow, then transported via secretory vesicles to the cell's exterior. So we're going to cover all of these pathways and the sorts of signalling that goes on in them in the next 2 weeks of lectures. So we've got a few things to think about, and we can cover these in the tutorials, but I'd like you to answer these questions, or attempt to, and then we can discuss your answers in the live tutorial next week. So proteins have nuclear import and export signals. How can proteins avoid constantly just shuttling backwards and forwards? So have a think about what proteins might do to prevent them just being randomly shuttled backwards and forwards in and out of the nucleus. Consider why endoplasmic reticulum proteins have a signal peptide on the N-terminus and not the C-terminus. There's a very clear reason why that might be. Endoplasmic reticulum proteins, and also some mitochondrial proteins as well, they enter as unfolded polypeptide chains. How might they change once they get into that destination? So particularly within the ER, what happens to proteins in the ER to make them functional? And then something else to think about what goes on in the nucleus. I want you to name 3 proteins that must enter the nucleus. I also want you to name 3 things that must be exported out of the nucleus. Okay, because this will help to explain the nuclear import-export lecture that's probably going to be next week. Okay, so we'll just discuss these in the live tutorial. So that's the end of part 1 of this week's recordings.